sticking around to one of the last sessions of the day. I appreciate it. Before I begin, I'd just like to remind us all that turtles are arguably, or probably not arguably, the most endangered group of vertebrates on the planet. And therefore, I think having a firm grasp of both population and species-level genetic diversity on these organisms is really necessary to ensure our conservation efforts are doing what they should for the survival of the group. So the species of interest in today's talk is going to be the flattened musk turtle, an extraordinary little turtle that looks almost as though a person with a heavy foot trod upon it. I think that pretty accurately describes this species. They are really small, large adults about 4 inches, and sometimes they can be dorsoventrally compressed to about 1 inch tall. These turtles are endemic to the upper Black Warrior River Basin in central Alabama, a pretty small distribution. And the rivers and creeks in this area are characterized by having this rocky substrate with large amounts of flagstone and even kind of these sublittoral low cliffs with all these cracks and crevices in them. And these turtles go retreat under these rocks and in these cracks and crevices. All right. So in the decades following the species description in 1955, people started noticing that populations were declining in number of turtles that were there. And to kind of expand on that, I used some historical survey, range-wide survey data, and some dynamic occupancy modeling, and really shown that these turtles are extirpated from between 36 and 52% of their historic range. And this extirpation is geographically cohesive with some watersheds in the distribution of this turtle completely lacking any individuals at all today. People hypothesize this is due to pollution, siltation filling these nice rock cracks the turtle likes to inhabit, disease, and overcollecting. In reality, I think all of these forces are leading to the decline of this species. In addition to population decline, morphology also suggests a narrow hybrid zone between the flattened musk turtle and a closely related loggerhead musk turtle, whose distribution is shown here in green and blue, depending on the subspecies we're talking about. And the narrow hybrid zone is here in kind of the lower end of the upper reaches of the Black Warrior River. Additionally, people hypothesize that this hybrid zone is actually relatively recent, being owed to the fact that the Black Warrior River has been undergoing a ton of anthropogenic change in the last 150 years, with all these locks and dams being put in place. And these locks and dams have decreased flow of the river, facilitating gene flow from minor into depressus. So this system's a little complicated, so hereafter I'm going to just kind of refer to depressus as depressus, and everywhere you see this nice ugly orange color and a flat turtle, we're talking about depressus. Minor is generally going to be this normal hump-shaped turtle in green or blue, depending on subspecies. And I think this system is best thought of as kind of this donut and donut hole, with depressus being the donut hole and kind of a single unidirectional river route to facilitate gene flow between these two species for a number of geographic reasons I can get into later, if you're interested. So first, when I started doing this study, I realized there's a lot of evidence for unidirectional mitochondrial introgression from minor into depressus. And so if you look at about 1,000 base pairs of the mitochondrial site B gene, you see a nice monophyletic, well-supported clade of Sternothrus depressus. There you can't see spores. These are all well-supported clades. Actually being closely related to other species in the genus, and a nice well-supported clade of minor individuals. And everywhere on this phylogeny where you see a green bar or a number of green bars, those are individuals of minor from the range of minor. And everywhere up here where we have this nice clade of minor haplotypes that you see orange, those are animals of depressus phenotype from the range of depressus that have mitochondrial haplotypes that are identical to that of minor from outside of the range. So either there's some sort of integration going on in the mitochondrial locus, or these things are sympatric, and I definitely shouldn't be working on turtles. And also really interestingly is these animals don't all come from this putative hypothesized hybrid zone, but you find these mitochondrial haplotypes of minor throughout the range of depressus, with only 3 sites I've sampled so far not having any individuals that have minor mitochondrial DNA. Good fun. So the major goals of my project, for the purpose of this talk, are to infer the extent of genomic integration between minor and depressus. And I think this is really important for a number of reasons, because I think it's well known hybridization can do anything from cause genomic extinction to genomic rescue, which can be really good or really bad. And so understanding what's going on in the system is important for conservation work. And also, it kind of allows us to ask questions about forces and mechanisms of hybridization and secondary contact on this river system that may be undergoing, anthropogenic change. And then also doing some work to understand population and watershed level diversity within depresses for setting conservation priority. And I won't have time to talk about that, unfortunately, but if you're curious, I'll gladly talk about it. So for this project, we've collected 3 rad-seq data, which is pretty similar to just a normal DD-RAD protocol with kind of some weird adapter stuff going on, kind of developed by Travis Glenn using XBA and EchoR1. We've sequenced 96 individuals on our Illumina NextSeq. I have actually a lot more than that, but I'm not talking about it. And 64 of those individuals are depresses. Demultiplex this stuff with stacks, de novo assembly, and SNP calling it PIRAD. And for a 90% complete matrix, a pretty conservative stacking and whatnot, we have a little over 1,300 loci, 6,000 SNPs, and 1,300 unlinked SNPs, which I'll hopefully use appropriately. So I think the first kind of nice thing to look at is look at population structure with these populations, and maybe that'll give us some sort of hint for the first sign of integration between these things. And what I have here is depressus ranged by watershed, with the putative hybrids here in the center, and then the 2 subspecies of minor kind of ranked by traditional taxonomy. And we can see at k equals 2 and k equals 3, there's no sign of any sort of integration from minor into depressus. And it actually looks like strong evidence for integration going the other direction. At k equals 4, which is the Ivano et al. Preferred number of clusters, we actually further subdivide minor with a lot of these individuals here making kind of their own cluster, and it looks like maybe there's some genomic leakage from these minor individuals which are proximate to depressus across the river into this putative hybrid zone. So far it seems like depressus is relatively pure, kind of nice, maybe a little bit of genomic leakage at the fall line where we expect there to be some integration. And maybe genetic assignment of minor is incongruent to an assigned subspecies, or even there's underdescribed diversity there, which could be kind of neat. I think the next kind of good thing to do is ask how these populations are related to each other. And so here's a Raxameltree using 1,300 loci. What we can see is depressus forms a nice monophyletic clade, which is well supported and sister to these weird kind of minor things that are falling out in structure. That clade is actually sister to reciprocally monophyletic and well-supported clades of both minor subspecies. So it looks like we have this weird thing going on here, and it's kind of curious as to what it is. Whatever it is, it's pretty closely related to depressus, or maybe actually depressus. All right. So what I then did is I wanted to look at genomic admixture index and genomic cline by location. So these are organized by river. Using Gompert and Berkeley's Introgression program. And so we have polarized this genomic cline and admixture index by pure depressus, pure minor. So these are things that do not have any sign of integration at all within them. And the first top 5 individuals here, so anytime you see kind of an orange, sorry, I should explain this better. These are all, each column represents an actual allele. Each row represents an actual individual. These are pure depressus from the range of depressus. These are all depressus from this putative hybrid zone. And you can see on this genomic cline, those are almost 100% depressus. And then we get into minor in the Black Warrior south of this, south of the expected kind of hybrid zone. You can see these things are actually all about 70 to 80% depressus, with this long kind of lingering tail of admixture. And this is generally a signature of a rapidly moving hybrid zone, which is leaving in its wake these lingering minor alleles. And so what it looks like is happening is there at one point was some sort of hybrid zone here, which has rapidly moved southward, leaving a wake of minor alleles behind it. Am I going way fast?
You have 5 minutes left.
Okay. Not too bad. All right. So in summary, I really think this system shows strong evidence for a mitochondrial sweep moving northwards from minor into depressus. And as this is not complete, it was probably pretty recent. And I think we have strong evidence for a rapidly moving southward hybrid zone from depressus into the range of minor. So depressus is actually expanding its range with this rapidly moving hybrid zone. And I think the next kind of logical step to do here is to actually incorporate models of river flow, both with and without kind of anthropogenic change, to figure out if this is a really recent thing happening in the last 150 years or so, or older, perhaps happening after like post-Pleistocene glaciation contact, to infer kind of what's going on. What I think is really nice is this is really good for depressus, because it really shows depressus within its whole range is pure, where it's still left, which is good, although there's some sort of mitochondrial leakage. If this really is a nice selective sweep, I don't think it's something we should perhaps be incredibly worried about, because there's some sort of pressure for it to be there. With that, I'll thank all these folks for discussion, these folks for funding, and letting me collect turtles on their land, and I'll take any questions.